Researchers have long been interested in the possibility of using stem cells to replace damaged heart tissue following a heart attack. Though transplanted stem cells develop into cardiomyocytes and integrate with the recipient tissue, preclinical studies and clinical trials have only shown limited improvements in cardiac function, perhaps because mechanical forces aren't transmitted properly between the immature stem cell-derived cardiomyocytes and the more mature host cells. The mechanical forces exchanged by cardiomyocytes can't be measured in vivo, however, so Yvonne Aratyn-Schaus, Francesco Pasqualini, and other members of Kit Parker's disease biophysics group at Harvard University developed a simplified in vitro system in which cardiomyocytes are paired together in two-cell microtissues.
So we have one cell that is a stem cell, derived cardiomyocyte, so it's more immature, it's weaker, it has all the characteristics of newly formed myocytes in the heart, and then a more mature myocyte that's going to represent the spare myocardium. And so when you put them together, and one is stronger than the other, we have this on-a-chip platform that allows us to study what happens at a number of different levels, in the structure, in the electromechanical coupling, and then in the actual contractile output.
Aratyn-Schaus et al. Used cardiomyocytes derived from either mouse embryonic stem cells or induced pluripotent stem cells. Because these myocytes are relatively immature, they contract more weakly than cardiomyocytes isolated from neonatal mice, which the researchers used as a proxy for the mature myocardial cells that survive following a heart attack. When stem cell-derived cardiomyocytes were paired with neonatal cells in microtissues, they formed normal-looking cell-cell junctions, containing proteins involved in both mechanical and electrical cell coupling.
And then, to be honest, we kind of expect the contractile architecture across the junction to be impaired. But we were surprised to find that at least, when you're just pairing a couple of cells, the contractile architecture looks really good pretty much immediately.
Aratyn-Schaus et al. Then found that stem cell-derived and neonatal cardiomyocytes were able to synchronize their calcium activity, and thus their contractions, when connected together in microtissues. However, resting calcium levels were reduced in stem cell-derived cardiomyocytes, and they were also lower in neonatal cells in heterogeneous cell pairs, potentially inhibiting contractility. The researchers therefore looked at the forces generated when weaker stem cell-derived cardiomyocytes were paired with stronger neonatal cells.
We originally had this idea that perhaps the stronger cell would provide an extra load that would, in a sense, train the weaker cell to become stronger over time. But what we found was actually that the extra force was dissipated away in the matrix, and so it wasn't really contributing to the overall contractile strength of the cell pair.
Specifically, the researchers found that, when cells of equal strength were paired together, they generated traction stresses at both ends of the microtissue. In heterogeneous cell pairs, however, the extra force generated by the stronger neonatal cardiomyocyte was transmitted to the extracellular matrix by focal adhesions that form near the cell-cell junctions.
And so, rather than having that structural coupling disrupted by the presence of a stronger cell that would just rip itself off the weaker cell, the cell actually lays down these adhesion sites at the junction that disperse the force away. And in the process, they sacrifice force transmission. And so you can see how that could actually impair cardiac cell therapy in vivo, because the force transmission between the old contractile material and the new contractile material is impaired. Then the overall performance of the organ might also be affected.
Of course, it will be important to know whether human cells behave similarly, but it is currently challenging to work with human cardiomyocytes in vitro.
So at the end of the day, we build a mathematical model that's based on frameworks, equations that have been used for the last 20 years to model both human and a variety of myocytes from animals, rodents, rabbits, and so on. And so really what the model does, it allows us to answer the question whether this mechanism was likely to be present in human cells. And it turns out that for you to see these junctional adhesions, the only thing that needs to change is some basic parameter into the sarcomere mechanics, like the isometric tension. If you reduce the isometric tension in one of the two cells, you are going to observe junctional adhesion and you're going to observe impairment of force transmission. So that was our way to sort of extrapolate our results into human cells.
The researchers' microtissue or muscle-on-a-chip technique should therefore help researchers develop ways to improve the mechanical coupling of stem cell-derived cardiomyocytes to surviving heart tissue. In the meantime, however, you can learn more about the coupling of primary and stem cell-derived cardiomyocytes in an in vitro model of cardiac cell therapy in the paper by Aratyn-Schaus and Pasqualini et al., published in the February 15, 2016 issue of the Journal of Cell Biology.